Good afternoon, welcome, buenas tardes. I'm Almudena Polo, and I belong to the EMEA Clinical Support Department of Vitrolife Group. I have experience in implement PGT programs in many clinics around the world, so I'm here to share with you a biopsy procedure and a tubing sample for PGT cases. So first of all, I want to thank you, all of you, the attendees here, but especially those who can't attend here in the Congress and they are watching in their homes. So I like to say that the biopsy procedure is a kind of multidisciplinary procedure because involve actually a IVF procedure, but as well involve a molecular procedure like tubing is. So first of all, I want to show you how to doing all the setup, all the preparation before starting actually the procedure. So we have two setups. One setup is for biopsy, actually, and the other is for tubing. So for biopsy, we have the needles in the microscope, inverted microscope with an octas laser that we have with micromanipulators and microinjectors. The needles are, of course, the holding that I recommend to use 120 micrometers because we're talking about blastocyst biopsy actually. So you have more surface to holding very well the blastocyst and avoid damage heat. And on the other hand, we have the biopsy needle that we use a flat one, and we use between 22 and 30 micrometers in the inner diameter. And we have here the biopsy dish. We use this square XC dish from Vitrolife. I can show you later. It consists in 2 drops between 20, 30 microliters of the biopsy media. We use GMAPS Plus that is allowed to operating outside the incubator actually, and avoiding changing the pH, get the sticky the sample into the dish, and of course covering by oil. So we recommend to use 1 dish per embryo because in blastocyst biopsy it's very important to get organized and avoid the sample sticky into the dish before they loading the sample into the tube. So we mark the biopsy dish with the number of the embryo, and if you have several patients, you can mark as well with the initials of the patient. So for biopsy, we have everything, and for tubing thing, we have just 2 parts of the setup. One part is going to be the washing part, we are going to wash actually the cells before loading, that is consisting the second part. For the loading part and for the washing part, we use the same buffer that we provide by iGenomics and we send to the clinics in order to we send a kit that it consists in has all the material you need actually for the tubing and for the shipment to the samples. So we have the washing disc that it has 4 lines. It has 4 droplets, sorry, about 10 microliters of the buffer. Each line corresponding to 1 sample. And you can cover with oil or not, up to you, depend on your skills manipulation and your optic of your stereoscope. And the other part, we have the rack. We send you as well in the kit. And we have several PCR tubes. I prefer to put extra tubes before doing the biopsy because, as you know, sometimes we don't know how many blastocysts we are going to biopsy, depend on the development of the blastocyst and depending maybe it's arrested. So I prefer to not mark the tubes, just to dispense with 2.5 microliters of the buffer loading. So once we have everything prepared, I'm going to proceed to the biopsy demonstration. So I have already here the blastocysts. We have some mice blastocysts. The first drop, it consists the one which we are going to wash the pipette and stabilize the pipette. And the second drop is actually the one you are using for the biopsy. So here we have several embryos. I'm going just to pick one. So here I have several embryos. Some of them they are very expanded. Some of them they are doing hatching already. So I'm going to pick this one, for example. I'm going to separate from the others just to have enough space. I'm going to go down with the biopsy pipette. And first thing I'm going to do is just move the embryo in order I can get the inner cell mass by 9 o'clock to avoid damage, because I have to take, of course, the trophoctoderm cells. So here we have the embryo. I'm going to move the embryo more or less like this. I put the several cells coming out through the biopsy pipettes, and once I get the position, I can move to the objective of the laser. Okay, so here, as you can see, we have the Octas laser. This is the home mode. I have to activate because it's very safe with some seconds of inactivation and inactivity. It's going to automatically going to inactivate. So I have to, with the key, activate. This is the home mode. In that mode, I can see actually the red target point here into the microscope. So you can see through the microscope, or you can see the screen, wherever you prefer. And we have another mode here that is the Navilase one. It's a very good software with you can control not just single pulse. You can do assisted hatching that we don't need now with that embryo, but you can use even assisted hatching not just for biopsy, just for zona thinning or pre-transfer embryos. And you can use the trophoctoderm mode that it consists in several multiples in order to cut the linkage between the cells. So I'm going through the home mode. Sorry. Yeah. And here I can move the target wherever I want. Even I can move the embryo, but I can move the target as well. So you don't need to move the whole system, the holding with the embryo, in order to get your target what you want. So in that point, I'm going to move to this because it's better to show you. Wait a minute. Okay. So in that point, I have the cells already outside. So I'm going to take the advantage of this. I'm going to touch the cells. And when I was touching the cells, I can start it aspirating. It consists in embracing the cell, not aspirate from far away. I need to get several cells, let for example between 5 and 8, and I can use the foot pedal actually to cut between the linkets into the cells. So I'm going to put all in the same focus because this is very important to have this in mind. For biopsy, you need to focus very well. And once I have, okay, and I get the biopsy. So here it will very quickly. So here you can see the embryo and the biopsy. In that moment, you can release the embryo. You can go in your laboratory. Of course, the first thing you have to do actually is return the embryo to the post-warming culture dish, or even the culture dish what you have. And now we are going to proceed to the tubing. So first of all, I have to say, for tubing, even for the setup, it's very important to clean everything, because the most important thing in the tubing is avoid DNA contamination. We have DNA in all our body, so please do it with gloves, of course, with ground and mask and everything you wear in your lab. But besides, you need to wear gloves because we are not in a molecular mode. Okay, so you have to clean every surface and every device you are going to get it inside the laminar flow hood. You have to imagine we have here a cabin with the decontamination product you have in your lab. You can use DNA away, you can use OSAFE, you can use Fermacidal. Even if you have some cabin outside the IVF lab, you can use alcohol even, okay? So it's very important, not just the surface, not just the stereoscope, all the devices you are going to get inside, okay? So this is a demonstration, so I don't have gloves, but I'm going to do it, the tubing procedure. So imagine I don't have the embryo anymore into the dish. I have my rack with the several tubes already dispensed, 2.5 microliters of the buffer we provide for my genomics. And here I show you the washing dish that is with the droplets I told you, from 10 microliters of buffer media as well. So the first of all we're going to do is washing the cells in these droplets, and then I'm going to load the cell into the tube. So now we know that that embryo is the embryo 1 for, let's say, AP, my name, patient. So I'm going to mark the PCR tube with the initials and with the number of the embryo. Once I have this, I took my device, hand device. I can use, with this buffer, you can use polycarbonate capillary, or even you can use glass capillary, whatever you want, because this buffer is completely designed for this. So now I'm going to pick the cells, the biopsy. There's plenty of embryos here. So imagine you have in your drop just the biopsy, because the embryo you are already stored. So I'm going to pick up the biopsy. I'm going to put in the first drop of the first line. Remember, each line consists in one sample. I'm going to release the biopsy, and the remaining liquid you can discard in any dish or even a lid you have. I'm going to move from this first drop to the second drop, release the sample, and again the remaining liquid discard into the lid you have or your discard dish. Again, I'm going to pick the biopsy. I'm going to put in the third drop and remind the discard liquid. In that moment, you have to be ready with your PCR tube that you can use your hand or you can use a lid just to support the tube and get you very easy the loading part. So I'm going to remove this. I'm taking the biopsy. I prefer just handling the tube with my hands, but you can do whatever you want, loading the cell, supporting the tube into the dish if you want. I have to focus the end of the tube. I entering with the capillary, and once I into the liquid, I starting to release until I see come out the cell. So now I can check in the fourth drop that there is, even you see the cells come out, you can check in the fourth drop if there's anything in the capillary. It's nothing there, okay? And a double check even is getting your tubes, putting like in the vertical way, and just try to focus and see the cell. Okay, and it's there. So close the tube, close your rack, and once you have all the samples, please store this into the freezer, in the -20 freezer. So that is all from my. Maybe I can take another biopsy, and I can show you for the people who are attending now. So I'm gonna try to do that. Again, you can do several approach for the biopsy. You can do the assisted hatching in the early day 5, and then you can perform the biopsy in day late 5 or even in day 6. Or there's some clinics that prefer to perform the biopsy, perform the assisted hatching, sorry, in day 3 and perform the biopsy in day 5 or day 6. Or if you don't have time for this, or you need to biopsy, for example, dowet embryos, you can use the pushing method, that it consists in opening the zona in the same moment that you are doing the biopsy. You can change the energy and the power of the laser even, and gently opening the zona until you get enough hole just to entering with the pipette. So here we have another one. I'm going through the objective of the laser. Remember, you can use with Navilase all the modes you have for assisted hatching, for multiples, even for moving your target. So again, I'm gonna touch the cells and starting aspirate. I recommend to embrace the cells recovering into the pipette without damage of stress the cells. When I have enough that I want, I can shoot. And I'm trying to show you how to do a flicking method, okay? The first one was pulling method because with 2 shots was enough to the cells cutting from the embryo, but now I can use the flicking method. I prefer when I shoot, for example, twice or maximum 3 times, go to the objective of 20, for example, to have more focus regarding the pipettes, because as I say, the focus is very, very important. But now to show you, I prefer to not move the objective. So here, as you see, I have very stabilized the pipette. The biopsy is not moving. Why is that? I forget to mention. Before I start in the biopsy and this procedure, I stabilize the pipette with some biopsy media inside the needle. Those microinjectors, they are from oil, but you can do with air as well. So once I have the biopsy into the pipette, I need to be sure that the pipette are in the same focus. Once I have the pipette, I can do like a little trace with the pipettes and the biopsy is cut. Okay, sometimes here is the biopsy. Sometimes the biopsy is very sticky into the pipette actually, so you can use a metal thick and just doing some touch here and the biopsy is going to release. So now I'm going to explain you again the tubing thing with the biopsy. Again, I'm coming into the cabin. Remember, in that point, use gloves and everything. Oh, sorry, I have to, yeah. Yeah. Sorry, in order you can see the tubing. Okay, now. So here is the biopsy. I'm going to pick once I pick the biopsy, I move to the washing dish. I put the biopsy in the first drop. Okay? And then I discard the remaining liquid. I pick the biopsy from the first drop and move to the second drop. Remain liquid discard. I'm picking again. go to the third drop. And once I have in the third drop, I can mark my second PCR tube. I can prepare the tube. Again, I can hold with my hands, or I can just leave here in the lid. Once I have the sample, I focus the bottom of the tube, get inside with the capillary, and release the sample. I can check in the fourth drop if there's nothing in the capillary actually. And the second check, just put vertically your tube, doing zoom, focus, and try to see the sample. This part sometimes is complicated, but it's like a second check. So close the tube, close your rack, and leave into the freezer till the shipment. So that's all from my side. Thank you very much.